Hello everyone. Today our lesson is on structure and functions of DNA. First of all, let's see why it's important for you all to study about DNA. There are many reasons. One reason is that abnormalities in human DNA can cause some oral diseases. One of the very common oral diseases resulting from DNA mutations is oral cancer. Another oral disease resulting from abnormalities in DNA is amylogenesis imperfecta. It's a rare disease, and the term amylogenesis means enamel formation. Imperfecta means not perfect. So the people having this disease have abnormal enamel formation. As you can see here, the enamel is discolored and we can see a lot of pits and grooves on the enamel. Sometimes DNA mutations can cause incomplete fusion of the two sides of the lip. This is something which happens during in utero development of the fetus, and we call this condition as cleft lip. It's not only the structure of human DNA, but also the structure of bacterial DNA can influence our oral health. These are the two most common oral diseases, dental caries and periodontitis. In the case of dental caries, the karyogenic bacteria produce acids and dissolve our teeth. And their DNA determine the structure and function of their acid-producing enzymes. Similarly, in the case of periodontitis, periodontopathic bacteria destroy our periodontal tissues using their protease enzymes. And it's their DNA which determine the structure and function of these proteases. This is how bacterial DNA can influence our oral health. Another reason for learning about DNA is that certain DNA viruses can cause oral diseases. For example, certain virulent strains of human papillomavirus such as HPV-16 can cause cancers in the oral cavity. And their strength or virulence is determined by their DNA. Then, one of the biggest challenges the whole world is facing today is connected with bacterial DNA. It is antibiotic resistance. Most of the disease-causing bacteria cannot be killed by the available antibiotics anymore. It's because bacteria have developed various mechanisms to get rid of the antibiotics and survive without any problem through DNA mutations. Another reason for learning about DNA is nowadays we use a lot of recombinant DNA vaccines to prevent diseases. For instance, it's compulsory for you all to have the hepatitis B vaccine before you start your clinicals, and that vaccine also is a recombinant DNA vaccine. And we use a lot of proteins made from recombinant DNA to treat diseases also. For example, recombinant human insulin is very frequently used to treat diabetes mellitus. In forensic dentistry, we use DNA to identify individuals. It is more accurate than using thumbprints in identification of people. Now, you know the importance of learning about DNA. So, let's learn about the location of DNA within human cells, the structure of DNA, organization of DNA in human cells, and also the organization of DNA in bacteria and viruses. Do you know where DNA is located in a human cell? Yes, most of the DNA is located within the nucleus, and a very small amount is located in mitochondria also. Now, let us look at the structure of DNA. A long time ago, two scientists described the structure of DNA. Do you know the names of those scientists? They are James Watson and Francis Crick. How did they describe the DNA structure? They described the DNA structure as a double helix consisting of two polynucleotide chains which are antiparallel and complementary to each other. Do you know why this structure is called as a double helix? Because it has two polynucleotide chains coiled around each other in a helical manner. Then why are these polynucleotide chains said to be antiparallel? Since the distance between these two chains is always constant, they are said to be parallel. And as you can see here, one polynucleotide chain is running from 5' end to the 3' end, and the other chain is running from 3' end to the 5' end. So they are opposite in direction. By the way, do you know what's meant by 5' and 3'? 5' means the fifth carbon atom of the sugar molecule is not engaged in a phosphodiester bond. That means it's free. And 3' means the third carbon atom of the sugar molecule is not engaged in a phosphodiester bond and it's free. So in the term antiparallel, anti means opposite. So antiparallel means the two polynucleotide chains are aligned in opposite direction but parallel to each other. When you look at these polynucleotide chains, you can see that they are polymers of deoxyribonucleotides. Each of these deoxyribonucleotides are made up of three components. One is the nitrogenous base, the other one is the deoxyribose sugar, and the third component is a phosphate group. As you can see here, alternating sugar phosphate molecules make up the backbone of the polynucleotide chain. And the nitrogenous bases are always directed towards the interior of the DNA double helix. These nitrogenous bases are of two types. They are purines and pyrimidines. Adenine and guanine are purines and thymine and cytosine are pyrimidines. As you can see here, hydrogen bonds are formed between these nitrogenous bases and stabilize the DNA double helix. Adenine always pairs with thymine with 2 hydrogen bonds, and guanine always pairs with cytosine with 3 hydrogen bonds. Therefore, adenine is said to be complementary to thymine and guanine is said to be complementary to cytosine. So the base sequence of the entire length of one polynucleotide chain is complementary to the base sequence of the other polynucleotide chain. Therefore, the 2 polynucleotide chains are said to be complementary to each other. Then, let's see how DNA is organized within our cells. As we discussed earlier, most of the DNA in our cells are found within the nucleus. Therefore, let's first discuss how nuclear DNA is organized. If we take out all the chromosomes in a single human cell and stretch them end to end, it will be around 2 meters long. But the approximate size of the nucleus in a human cell is around 2 to 3 micrometers. So how is it possible to put 2 meters long DNA into a 2 to 3 micrometer sized tiny nucleus? It is through very tight packing. So who helps DNA to pack so tightly? They are the proteins. There are specialized proteins to bind with DNA and fold them into higher and higher levels of organization and make chromatin and finally the chromosomes. Then, what is the nature of these DNA binding proteins? Are they acidic, basic, or neutral? As the name implies, DNA are acidic, deoxyribonucleic acids. So in order to bind with them effectively, the DNA binding protein should be definitely basic. And these proteins are classified into 2 groups. One is histone proteins and the other one is non-histone proteins. At the very basic level of DNA organization, mainly the histone proteins help DNA packing. At the higher levels of organization, mainly the non-histone proteins help DNA packing. At the very basic level of nuclear DNA organization, histone proteins bind with DNA to give a beads on a string appearance. So these are the beads and this is the string. These beads are called as nucleosomes, and each of these nucleosomes are made up of 8 histone molecules surrounded by the DNA. These histones can be modified They can undergo various types of modifications depending on our diet and how frequently we exercise, and also depending on the environment and also the nature of our mind. Two of the most common modifications in these histones are acetylation and methylation. These modifications can affect the coiling and uncoiling of the DNA bound to those histones. Especially uncoiling of this DNA is very important at the time of DNA replication and gene expression. That is, processing of the genes in DNA to make RNA molecules or proteins. So after finishing these processes, the DNA should uncoil once again. A long time ago, DNA was considered a supermolecule which can determine our entire structure and functions. So we talked only about genetics. But now we know that many other factors can influence the structure and functions of our body without changing our DNA. This is epigenetics. Many epigenetic factors such as exercise, nutritional factors, environmental factors and emotional health can influence histone modifications and change the expression of our genes without altering our DNA or the genes. Now you have learned how DNA is condensed and organized within our nuclei. In this picture, I have shown you two cells. This is cell A and this is cell B. Can you tell me the difference between the nuclear DNA in these two cells? In cell A, this is the nucleus and its DNA is found in the form of chromatin. And in cell B, this is the nucleus and its DNA is found in the form of chromosomes. If we want to see this type of very clear chromosomes, what we have to do is we have to allow the cells to divide and stop the cell division at the metaphase and observe under the light microscope, because metaphase of the cell division is the time where the cellular DNA will be maximally condensed. Every species has a specific number and appearance of chromosomes. It is called the karyotype of that particular species. In the case of human, we have 23 pairs of chromosomes and out of them 22 pairs are autosomes and the remaining pair is the sex chromosomes. As you can see in this picture, different chromosomes have different lengths and different shapes. So the number, the size and the shapes are collectively called as the karyotype of that particular. By looking at the karyotype, we can identify certain genetic diseases. Diseases arising due to increase in the number of chromosomes or reduction in the number of chromosomes, or else increment or reduction of the chromosomal length can be diagnosed by looking at the karyotype. Now, can you tell me the abnormality seen in this particular karyotype? It is chromosomal trisomy at the 25th first position. This is how we write the karyotype. First we have to write the total number of chromosomes, and then the sex of the individual, and then the specific location where the additional chromosome is found. And what is the disease condition arising due to this abnormal karyotype? It is Down's syndrome. Now, let's look at the organization of DNA in our mitochondria. Our mitochondrial DNA also are double helixes as explained by Watson and Crick. But they are condensed into circular molecules. Their condensation also is supported by proteins, but unlike the nuclear DNA, mitochondrial DNA condensation is entirely supported by non-histone proteins. This mitochondrial DNA arrangement, that is the size, the shape and condensation, is very similar to the arrangement of DNA in bacteria. That is why the scientists say that our mitochondria have evolved from bacteria. When we take all the DNA in our nuclei and mitochondria together, we call it as the human genome. DNA in the nuclei belong to the nuclear genome, and the DNA in mitochondria belong to the mitochondrial genome. These two genomes are totally different. As you can see here, the size of the nuclear genome is very large compared to the mitochondrial genome. And the DNA in the nuclear genome are arranged into linear shaped chromosomes. And the DNA in the mitochondrial genome are arranged into circular shaped molecules. And the most important and the most interesting difference between these two genomes is that only 2% of the areas of this large nuclear genome code for proteins or RNA molecules. In the mitochondrial genome, most of the DNA code for proteins. They are the mitochondrial proteins and mitochondrial RNA molecules. Now, let us quickly look at the organization of DNA in bacteria and viruses. Most of the DNA in bacteria are located in the bacterial chromosome. In addition, they have some extra chromosomal DNA also. They are called plasmids. They are very small and circular and responsible for carrying the antibiotic resistance. When we look at the organization of DNA in DNA viruses, we can identify two types of organization. In some viruses, the DNA molecules are organized into double-stranded molecules, and in certain other viruses, the DNA is organized into single-stranded molecules. In dentistry, we generally don't encounter diseases caused by single-stranded DNA viruses, but there are some common diseases which are caused by double-stranded DNA viruses. One of those viruses is human papilloma virus, which can cause oral cancer, and the other one is Epstein-Barr virus. So that brings us to the end of today's lesson, and we have learned about the structure of DNA and how they are organized in our cells, and also the importance of learning about DNA. In our next lesson, I will discuss about the genes, the functional unit of DNA.